Hey biology students, this is the last notes for protein synthesis. Thanks for doing this podcast, Genetic Technology. Remember to pause if you need to. Let's go. So also, genetic technology will sometimes be called biotechnology or genetic engineering, but all three of those are going to just be referring to technology used to manipulate DNA. Recombinant DNA, which is manipulated through technology, meaning biotechnology, refers to DNA from two different sources. An organism will be called transgenic if it received genes from other species, meaning transgenic organisms have recombinant DNA. So for instance, broccoli, scientists have taken genes from broccoli and genes from cauliflower and combined them in another organism, and they made a weird thing called broccoli-flour. Now, do we see broccoli-flour really in the stores? No, because it's expensive to make and, well, kind of freaked people out. And we'll talk about some of the ethics of this gene technology or biotechnology later. Another example of a genetically modified organism or transgenic organism is that we can take the genes from an organism, right, and they can be replicated in another organism. For instance, we can take a human cell and we can take some of the genes, one particular gene out of it, like the insulin gene to make the insulin protein, and we can put it together using gene technology, and we can combine it with the weird bacterial circular gene that you'll learn more about in IB biology. This is the majority of the bacterial DNA called a plasmid. It's just circular. And if we combine them and put it back into the bacteria, this bacteria will multiply and all of its daughter cells and the original bacteria will do protein synthesis, transcription and translation, and they'll make the protein insulin. Now, diabetic people don't have a working protein insulin, so we can kind of take all that insulin out of these bacterial cells and we can make medicine for diabetic people who can't make their own. And we wouldn't have been able to do this just by taking insulin from humans. They don't make enough to share with others. So by putting in bacteria and having them produce a lot of it and using their own protein synthesis machinery in their cells, we can make lots of insulin. It's pretty amazing. Other genetic technology, CSI stuff, like DNA fingerprinting. So humans have fingerprints, right? Look at your own fingers. They have those rings, and all of us have different rings on our fingers unless we're identical twins, right? And we can use those to identify different people. Well, interestingly enough, other than 99% of our genome or all of our DNA, humans have at least 1% that's slightly different. And that 1% of our DNA for all of us makes us individuals other than identical twins. So we're going to harness these differences in our DNA to analyze things called DNA fingerprints. And they're going to look like this picture to the right. Let's learn how we make them. So a DNA fingerprint is going to be made through DNA fragments. So we got to chop up the DNA, and that's done fast, and that means we need an enzyme. So enzymes, these particular types of enzymes you'll learn more about in IB biology, cut DNA at specific sequences. So here's an example. So let's say we, within our genome, have at least 100 times this little sequence of 3 C's and then 3 G's. All right, and so the enzyme will find those sequences within our genome and some of our cells, right? And every time they see this kind of in-between spot with 3 C's and 3 G's, right in between the C and the G, they'll chop it in half, right? And now maybe I have 100 of those sequences, but maybe you only have about 90, and they're all in slightly different locations within your genome than me. So our chopped up DNA sizes are going to be a little different than each other. So those different sizes are going to be able to show us the difference between you and me. And if I separate those different size fragments by their size, I could figure out which of them is me and which of them is you, because no one has identical DNA. So that's what a DNA fingerprint is. We're going to do the basics through the podcast, but we're going to learn more details in class together, okay? So how do we then separate those fragments? Well, we're going to use electricity through an electrical current, and it's going to be this technique called gel electrophoresis, and you'll learn a lot about that in a couple of years when you take IB biology. But it's going to use an electrical current to send the DNA fragments through this gel-like structure, and it's going to separate by size. If you're really excited and curious how they do it, because this is exactly what they do at CSI labs, you can watch this really neat animation by typing this into your browser. But it can do a lot of neat things, like compare the relationship between different types of organisms. It can do paternity tests. It can solve crime scenes. So we're going to do a couple examples through this podcast, but really what you really need to do is, if you want to learn more, go to this cool website, because I can't do it as well. So we're going to look at some pictures of these fingerprints, but in class we're going to learn the details of how to analyze them. So here's some examples of a use of a DNA fingerprint. We can use it to figure out who the father is if we don't necessarily know the father of a child, which is called a paternity test. So here's an example picture. Most of the time we look at the rows of the DNA fragments to analyze these. We'll learn about how to do this in class, and I'll show you this exact figure in class. We can also analyze crime scene DNA data, looking at the DNA at the crime scene and compare it to different suspects. Again, we would look at the different rows and see which DNA looks more similar. If you want to try it on your own, try to figure out which suspect did it. I can give you a hint. It's going to be the one that has the most similar DNA to lane 2. All right, we'll do it in class together if you want to try it. Don't try to draw it. All right, let's learn about some of the products and uses of genetic engineering. Well, there's a big controversy right now in the news about genetically modified food, which could help maybe make more food for the world, but there's a lot of risks. Sometimes there might be allergens in this transgenic food. If I add a fish gene into a tomato and people who are allergic to fish eat the tomato, do they get sick? We're not really sure. Hopefully not. And we're not really sure what kind of consequences might come from this food going into the wild. But there's a lot of benefits. Maybe this food will be drought resistant and could grow in the desert where food currently doesn't really grow, and maybe it'll feed all the hungry people in the planet, because about 1/3 of the planet is not getting enough nutrients in food. Other things, we can investigate human genetics and diseases. The major project that did this was called the Human Genome Project, that started in the 90s, and they made a lot of breakthroughs in the 2000s, and what that was an international study to know all that you could possibly try to know about the DNA of the average human, because we have a 99% common DNA for all humans. So they wanted to know every single gene that humans had and what their proteins for those genes coded, and what each of the functions were for that. And that's kind of actually what Virginia Tech's working towards for Arabidopsis, right? So they're investigating a particular gene in Arabidopsis to learn about its function, and they're made a mutant, and they're trying to understand how that gene works by knowing how the mutant reacts to different environmental conditions, and you guys are helping with that. Other things, genetic therapy, which is trying to cure a disease that's related to a typo or mutation in the DNA that might be making a defective protein. And what they'll try to do is they'll try to either change the gene itself, which is hard because you would have to do it in all the affected cells, and we have lots of cells, or trying to add a working protein. This is very expensive and hasn't been super successful, but it's kind of cutting edge and really interesting. We could also screen for genetic diseases, and what that would look like would be maybe looking at the DNA of parents and trying to see what the DNA might look like in the children. So do those children, are they going to get a genetic disease? We're going to learn more about that and some of the genetic diseases related to cancer later in the upcoming units. But all those things are related to ethical issues. Do we think it's right or wrong to know what kind of genetic diseases you might get in the future? Do you think it's right or wrong to manipulate the genetics by putting, for instance, a human ear on this mouse, but this human ear is going to be given to someone who had their ear burned off through an accident in the Afghan war, and they didn't have a working ear at the time anymore? But we don't always know the consequences. What's the consequences of our food genetic modification, the consequences of manipulating even human genes? So here's some ethical things to consider. Maybe if we modify organisms, we might accidentally produce harmful ones, bacteria that might eventually hurt us. A lot of times we knowing the genetic diseases before organisms born, like a human, it might be complicated to know if they should be born, the human should come into the world in the first place. This is hugely controversial. Should we even modify humans' genes in the first place? What if you could go and tell a doctor, oh, I want my child to have red hair and to have adorable little freckles, but is that fair? Is what will happen to all of our human diversity? It's a lot of complicated things to consider. Genetic screening and these changes will eventually be very expensive, and they already are. Is it going to be okay if people who are struggling to make ends meet to even eat, they're not going to be able to do these really amazing things or somewhat scary stuff? And hopefully we're not using this technology to make weapons, all right? I mean, this is a silly picture, but we wouldn't want the technology to be used to hurt other people. So we're going to talk about some of these things in class, and we're going to especially learn about DNA fingerprinting. Great job today, guys. I hope you think about some of these ethical issues, because eventually you'll be someone who's voting on them in the future, because this is your world and you're going to have to decide how we should have the world be in the future. Thanks, guys, for doing the podcast.